Good afternoon, everyone. I'm Amy Hajari Case, Senior Medical Advisor for Education and Awareness at the Foundation. And I want to welcome you all to our final session of the live session of the 2021 PFF Summit. It's been an amazing week of education, support, and community, and I'm excited that we'll be wrapping up this afternoon with a discussion of therapies under investigation for pulmonary fibrosis. Our faculty for this topic is Dr. Toby Maher, Director of the ILD program at Keck School of Medicine at the University of Southern California. We'll hear first from Dr. Maher and then take questions from our audience.
Hi, everybody. I'm Toby Maher. I'm Professor of Medicine and Director of the Interstitial Lung Disease Program at the University of Southern California in Los Angeles. It's a pleasure to be joining you all today to talk about the current landscape of active pulmonary fibrosis-related clinical trials. Thank you very much to the Pulmonary Fibrosis Foundation and the organizers for inviting me to speak on what I think is a very important topic. So just to give a little bit of background on myself, my accent perhaps gives away that I'm not a native of Los Angeles. In actual fact, I've spent most of my career working in London at the Royal Brompton Hospital, where I followed in the footsteps of the lady in the center of this slide, Dame Margaret Turner Warwick. She set up the interstitial lung disease unit at the Brompton back in the 1960s. And so I was in the fortunate position of working with a huge number of patients with pulmonary fibrosis, all of whom were very engaged in clinical trials. And so I spent a lot of the last 15 years running clinical trials there. And I've subsequently transitioned to Los Angeles, where I'm now working at the University of Southern California to build a similar IPF and ILD clinical service and clinical trials unit. So these are my disclosures. So this is what I'm going to go through in the talk. I thought I would start by giving some background and discussing really why it is we perform clinical trials, and just give a brief overview of the process of clinical trial development of new drugs, because that then helps to put in context the discussion about new drugs that we are trying to develop for idiopathic pulmonary fibrosis and other forms of pulmonary fibrosis. And then I'll talk through the main drugs in development, and include in that some discussion about other approaches that are being taken beyond just treating fibrosis. So drug development can seem quite complicated from the outside. It's something that the pharmaceutical companies spend billions of dollars on each year, and which is a very heavily regulated process. In the United States, we have the FDA, who oversee both clinical trial delivery, and they also assess the results of clinical trials before approving new medications for use in clinical practice. And so there is a whole process that has been developed for taking new drugs from discovery through to showing that they work in humans, that they are effective in patients, and then allowing them to be approved for treatment in clinical practice. And this approach to drug development breaks down into a number of phases. On the left-hand side of the chart, we have phase 1, and before that, we have preclinical development. Preclinical development is really coming up with the idea for a new drug, testing it in cell cultures in a laboratory, testing it in animals to make some assessment of safety. And only when all that has been done does it move into phase 1 trials, which are the first time that the drug is put into humans. And then as we become more confident about the effect of the drug, we move through different phases until after the end of phase 3, when we hope that the drug will be approved. Just to go through this in a little bit more detail, phase 1 trials are really only undertaken after animal safety studies have been completed. Certainly when it comes to pulmonary fibrosis, phase 1 trials always tend to involve healthy volunteers. In some cancer studies, drugs go straight into patients in the first instance, but that's not happened in pulmonary fibrosis. There tend to be 2 different parts to a phase 1 trial. So in the first part, individual subjects are given just one dose of the treatment and monitored to make sure there are no unexpected or adverse effects. And when that's been completed in a number of dose ranges, there is then a sort of multiple dose a part of the study where individuals will take repeated dosing over a number of days, usually 2 to 4 weeks. And again, lots of measurements are made to make sure that the drugs don't have unexpected effects or cause unanticipated side effects. And the real purpose of these phase 1 studies is to make sure that the drug is generally safe, but also to give the investigators the chance just to explore the characteristics of the drug in more detail. Before a phase 1 trial, a lot of the information that has been developed has either been developed in cell cultures or animals. And so although there's a broad understanding of how a new drug might behave, there's always a little bit of uncertainty about how it will behave in human beings. And so the main aim of this study is to make sure that the drug behaves in exactly the way that was predicted in the earlier laboratory experiments. And these studies are important because occasionally surprises happen. This study on this slide is an example. It was highly publicized at the time of a phase 1 study that went wrong. So this was an antibody study. They gave the drug to 7 healthy volunteers simultaneously, and all of them developed a so-called cytokine storm, which is what we've seen over the last 18 months in patients with severe COVID. And these patients all became very sick, but fortunately survived the experience. But it was an important lesson in the very essence of doing phase 1 trials, which is to make sure a drug is safe before it goes into patients with diseases. Once we've completed a phase 1 trial and it's been agreed that a drug is safe, we move into phase 2. And phase 2 often splits into 2 parts, so-called phase 2A and phase 2B. Phase 2 tends to be the first time that a drug is tested in people with the disease of interest. So obviously, in the context we're discussing today, that would be people with either idiopathic pulmonary fibrosis or people with other forms of fibrotic lung disease. As I say, there's this separation into part 2A and part 2B. Part 2A studies tend to be very short and intensive, and they're very similar to the phase 1 studies. The goal of these is to make sure that the drug behaves in the way that was anticipated when it was designed. The phase 2B studies tend to be somewhat longer, and the goal of these studies is to make sure the drug has the anticipated effect. So if we're trying to design an anti-fibrotic drug for people with pulmonary fibrosis, in a phase 2B study, we want to make sure that the drug is actually having an effect on measures of pulmonary fibrosis. So normally that would be lung function measures such as forced vital capacity. And we would want to see over the course of the study that the drug was having an influence on change in fibrosis and lung function over time. This is an example of a phase 2A study that we conducted a few years ago. In this sort of study, we had 4 groups of patients with 4 patients in each group. Each group of patients received a different dose of the medication, and we very intensively monitored them. We did so-called PET scans, which are scans that look at metabolic activity in the lung, and we did repeated bronchoscopy to look at the cellular behavior in the lung as well. And this study told us that the drug acted in the way that was anticipated and gave us good information about the optimal dose of treatment to be used. This, by contrast, is a phase 2B study. This was the original clinical trial with nintedanib, one of the anti-fibrotic drugs we now use in clinical practice. And in this trial, the investigators tested 4 different dosing regimens. So they gave different groups of patients either placebo, or they gave them 1 of 4 different doses of drug. And you can see on the graph, the different colored lines represent the different dosing groups. And this study gave important information suggesting that the highest dose, 150 milligrams twice a day, was the dose that had the greatest effect on slowing pulmonary fibrosis. And then after we do our phase 2B study, we move on to phase 3 trials. These are the most heavily regulated of the trials. These are the trials that will be presented to the regulatory agencies. So the FDA, the Federal Drug Administration in the United States, or the European Medicines Agency in the European Union. And so these studies have to be designed to show that the drug actually has the anticipated effect. So again, in pulmonary fibrosis, that would be to show that a drug is anti-fibrotic in action. As a consequence, because of this need to show effect, these tend to be longer studies with far many more participants. Regulatory agencies usually want to see that the drug has worked in more than one trial. So normally there will be at least 2 phase 3 studies done with a new drug to prove to the regulators that things are effective. And these are the positive phase 3 trials that we've seen in idiopathic pulmonary fibrosis. So we had the ASCEND trial and the earlier capacity trials for pirfenidone, and we had the Impulsis trials for nintedanib. And in these studies, patients were studied for a whole year. The primary endpoint measure, the measure that showed the drug worked, was forced vital capacity, so a measurement of lung function. And all of these studies required between 500 and 600 participants in each trial. So they were pretty large undertakings. And then I briefly mentioned phase 4 trials. Phase 4 trials are done once a drug has been approved and is available in clinical practice. And these are normally done to give us better information about how a drug might work in specific subgroups of patients, or they can be done to look at combinations of treatments together, or they can be done to look, for instance, for biomarkers. And this is just an example of a phase 4 study that we ran using nintedanib, where we were very specifically looking for blood measures that would predict effectiveness of treatment in patients. So what else do we need to think about in clinical trials? Well, there is the issue of placebo. So when we do studies, almost always we use a placebo as well. So this is a dummy treatment that looks exactly the same as the treatment that's under study. And the reason we do that is to make sure that findings that we think can be attributed to the drug really are, because often when people are in clinical trials, they will tend to feel better. And those improvements in how they feel can mistakenly be ascribed to drug effect when it's simply the effect of being in the trial. And so having a placebo group allows us to tease out both the beneficial effects of the drug, but it also allows us to identify the negative consequences of the drug as well, the side effects. And these studies are important because we've had many trials where there's been a lot of hope and expectation, where ultimately it's been proven that the drug being tested is actually worse than no treatment at all. People often consider it a raw deal to end up on the placebo, but sometimes in clinical trials, it can be the better option. It's also worth saying that clinical trials take up a lot of time, so people have to be invested in them. This diagram is a schematic of a recently conducted phase 3 study, and each of the vertical lines on the graph represents a trial visit. So you can see over 52 weeks, there is an expectation that patients are going to come up to the study center at least 12 times. And so it is quite a commitment for people to be in clinical trials. We've talked about placebo drugs. Often with phase 3 trials, once patients have completed the trial itself, there is often the opportunity for patients to continue on open label treatment. So if you're in a trial with a drug that proves to be effective, often people get early access to treatments that will take another 12 to 18 months to get approved before the end of the study. And this is just, again, an example. These were the Impulsis trials in IPF. Everybody who was in the trial finished the trial after 52 weeks, and everybody was given the opportunity to switch on to open label nintedanib. So everybody who'd been on nintedanib carried it on. People who were on placebo got switched at that point onto nintedanib. So everybody got access to treatment afterwards. And the reason for doing these studies is not so much to reward people for their participation in the trial. It is instead to get much longer detailed information about the safety and effectiveness of the drugs. And then there's a lot that goes into running clinical trials. Of course, we realize that by exposing oneself to an experimental medicine, there's always the risk of things not working, or even the risk of developing side effects. So there are a lot of things put in place to make sure that participants are protected. And so the site that's running the trial will have responsibility and oversight for individuals that they're looking after. The sponsor, which is to say whoever it is that's funding the study, will visit sites regularly to make sure things are being conducted appropriately. In the United States, the FDA will periodically visit sites and undertake inspections to make sure that things are being conducted safely and appropriately. And most trials also have something called a data safety monitoring board, which is an external group of experts who periodically look at the data generated by the trial to make sure that there is no harm coming to participants. And occasionally we see trials stopped because the Data Safety Monitoring Board is concerned that the drug may be having a negative effect on participants. And then this is another challenge of running trials. So this was my trials team when I was back in London. And this is really just to demonstrate that running trials is quite a big undertaking. At any one time, I would have about 15 people helping me with running trials, including research nurses, laboratory technicians, lung function technicians, other doctors who helped assist with running it day to day. You can see that they get thirsty after a long day of work, but it just shows that in each individual trial center, there's a lot of effort that goes into running and delivering trials. And that does come at a cost. It's worth saying that trials are very large and complex. They require multiple sites. So your typical phase 3 study in pulmonary fibrosis will require 120 or more different hospitals to participate. And that will cost the pharmaceutical companies that tend to run these studies anywhere in excess of $250 million to deliver trials. So these are massive undertakings. So that's the background to clinical trials. The question is, where are we now in pulmonary fibrosis? Well, over the last 10 years, we've made some important strides. And perhaps the first and most important study that we saw published in the last decade was this one. And this was funded by the NIH in the US and was delivered by the IPF Clinical Research Network. And this was a study looking at what was the recommended best treatment for IPF at the time, which was considered to be a combination of steroids and azathioprine, plus or minus the antioxidant N-acetylcysteine. And for the previous 10 years, the combination of prednisolone and azathioprine had been the recommended treatment in the IPF guidelines. And actually, it was only when this study was conducted that it became clear that prednisone and azathioprine together were actually harmful for patients with IPF, that we actually saw an excess of deaths and hospitalizations. And really, this study proved the importance of running clinical trials before we adopt treatments into clinical practice. And so this first study really led to the discontinuation of high doses of corticosteroids and azathioprine as a treatment for idiopathic pulmonary fibrosis. The next set of important trials were the phase 3 trials for both pirfenidone and nintedanib. And as I've already shown you, both of these drugs were shown to be effective, with treatment slowing the rate of disease decline by about 50%. And 7 years on from the approval of those drugs, we've now got real-world data that shows that active treatment with antifibrotic drugs improves survival for patients with a diagnosis of idiopathic pulmonary fibrosis. And this was a study we did trying to estimate how much extra survival it was getting for patients. And it would seem that effective treatment, and for patients who can stay on treatment, life expectancy is improved by anywhere between 2 to 3 years. So really quite a significant effect. However, what we also saw in this study was that compared to age-matched individuals from the general population, even with treatment, patients with pulmonary fibrosis lose life expectancy. And as a consequence, we still need more and better drugs if we're going to completely restore normal life expectancy to people with pulmonary fibrosis. The next development we've seen is the use of antifibrotic drugs in diseases other than idiopathic pulmonary fibrosis. So this was a trial of nintedanib in patients with the autoimmune condition, systemic sclerosis. This is a condition where we see multi-organ problems due to autoimmune injury. And about 3/4 of patients with this disease have pulmonary fibrosis. And we were able to show that nintedanib, as it is in IPF, is effective in slowing disease decline. And as a consequence, nintedanib has now been approved for patients with systemic sclerosis-associated ILD. And then the next development we saw, again with nintedanib, was the use of an antifibrotic drug in patients with any form of pulmonary fibrosis. So this trial, the INBUILD trial, was conducted in patients who had pulmonary fibrosis that was due to any condition apart from idiopathic pulmonary fibrosis. And what this trial showed was two things. One important thing was that it showed that pulmonary fibrosis that's not due to IPF is a big problem that causes worsening of lung function over time. But it also showed that antifibrotic therapy can be effective in this group of patients in slowing disease decline. So showing us that treatment of fibrosis is both important and possible across a wide range of fibrotic lung diseases. So what of the future? Well, we increasingly recognize what it is that leads to the development of pulmonary fibrosis, and our improved understanding of the mechanisms that cause fibrosis is leading us to better identify new treatments. And what we recognize about the causes of fibrosis is that there are three main factors that seem to contribute to people developing fibrosis in the first place. One is genetic susceptibility. So we recognize that abnormalities in a range of genes related either to the aging of cells in the lung or to the lung's ability to protect itself make people more likely to develop fibrosis. We recognize that getting old contributes to the development of the disease. We very rarely see pulmonary fibrosis in young people. We very frequently see it in people over the ages of 60 or 70. And then the third important factor is injury or damage to the lung. Most often in idiopathic pulmonary fibrosis, that's due to environmental exposure and injury. That can be breathing in dust or smoke or pollution or viruses or bacteria. In people with autoimmune disease, that injury is often caused by that individual's own immune system attacking the cells in the lung. Once you've got those three factors that trigger the development of fibrosis, what we then recognize happens is that we get activation of pathways involved in the normal wound healing response. So the same pathways that would heal your skin after injury are the same pathways that become active in the fibrotic lung and ultimately cause the laying down of scar tissue. At a molecular level, we see an imbalance between mediators that drive the development of fibrosis and those that would normally help to prevent it. And that favors the activation of cells that make the fibrotic tissue. And that's what ultimately causes the scarring that characterizes pulmonary fibrosis. And importantly, the other thing that we see is that the process becomes self-perpetuating, that as the lung becomes scarred and stiffer and the nature of the makeup of the lung changes, those factors in themselves have an effect on driving more fibrosis. And so by recognizing the pathways involved in the development of fibrosis, that allows us to identify different ways in which we might be able to treat fibrosis. This little schematic here is just trying to illustrate the different parts of the fibrotic pathway. So we have the injury and damage to the lung. We have activation of pathways, including inflammatory cells, clotting cascades, and then the different cell types involved, the epithelium and the fibroblasts being the structural cells that make the scar tissue. And then in the boxes here, we have the different things that are being targeted to try and prevent fibrosis. And this is perhaps not an exhaustive list. The challenge with drug development in IPF is that it's changing very rapidly. And there are probably 30 or 40 different compounds in development. And many of them tend to be very different, targeting different pathways. But this schematic gives a broad overview of many of the main pathways that are being targeted. The bolded pathways are the ones where we've seen the greatest drug development. The pale pathways are ones where we've seen negative trials, and probably we won't see drugs developed for those. And the intermediate font are other pathways that are currently being tested in clinical trials. So for the rest of the talk, I'm mainly just going to focus on those trials that are most advanced. I could spend the next 10, 15 minutes talking about 30 or 40 different treatments, but I thought it'd be more fruitful to focus on a smaller number of drugs, and those drugs that are in the furthest stages of clinical development. And you'll see that the first drug on there I've crossed out because we know that the trial has recently failed. But I thought it worth talking about it nonetheless, just because there are some lessons we can learn from it. So I'll just go through each of these. So the first drug there is zirotixistat, or as it was known at the time, GLPG 1690. And this inhibits something called autotaxin. Autotaxin is a protein that signals to cells various things following injury. And it drives cellular responses to injury and ultimately drives the production of collagen and scar tissue. And there's a lot of good data from basic science and cellular models to show that autotaxin plays a key role in the development and evolution of fibrosis. And if we look in animal models, blocking autotaxin can help to prevent fibrosis. And so about 5 or 6 years ago now, we started off by running a small trial of this autotaxin inhibitor in just under 25, I think it was 23 patients with idiopathic pulmonary fibrosis. Patients were either given the active drug or they were given placebo. And because this was a phase 2A study, the main thing that we were looking at was whether the drug had the anticipated effect. And so we were looking for the downregulation of other proteins in the blood that correspond with inhibition of autotaxin. And so what we see on this graph is the placebo group in black and the treatment group in yellow. And what we saw was that the drug did what we expected it to, which is to say that it blocked autotaxin pathways. But interestingly, what we also saw at the same time, even in a study with such small numbers, was that there appeared to be a benefit in terms of lung function in the patients given the active drug. So here you can see in red the patients given treatment, in blue the patients given placebo, and you can see over the 12 weeks of the study that there is some separation of the lines between the two groups, suggesting that active treatment may have been associated with a slowing in lung function. And that led on to the phase 3 clinical trial program. So this was 2 parallel studies, the goal being to recruit 750 patients into each one. So a massive undertaking and a big step up from a 23 patient trial to a 750 patient trial. Unfortunately, the study was terminated about 6 months ago because the Data Safety Monitoring Committee no longer felt that there was a sufficient benefit to patients, and they were concerned that there may even be some sign of harm. The challenge, of course, was that the study was running through COVID, and so there were difficulties in interpreting the effect of COVID on patients with pulmonary fibrosis. We've yet to see the published data, but unfortunately it means that zirotixistat is not going to become a treatment for IPF. There is, however, still interest in blocking autotaxin, and so there are a number of other trials going on at earlier stages of development with different autotaxin or so-called LPA antagonists, which again, the hope is may be more effective than this one was. The next pathway that has been looked at is the so-called CTGF pathway, which is connective tissue growth factor. Connective tissue growth factor is another protein that signals to cells to stimulate the development of fibrotic tissue. In IPF, we know that there's an excess of CTGF, And we know that that can be blocked using antibodies. And so that's led to the development of a drug that was initially called FG3019, has later been given the name of pamrevlumab. This is a monoclonal antibody. So antibodies are things normally produced by the body to fight infection. But there are a range of treatments now used for different diseases where the power of antibodies has been harnessed to block specific proteins in the body. And so pamrevlumab is a monoclonal antibody that blocks CTGF. And so there was an initial open label trial published in 2017. So an open label trial is a trial without placebo where all patients know what treatment they're getting. That showed some signs of benefit. And so it led on to a double-blind placebo-controlled trial, the results of which were published about a year ago. And in this longer, larger study, so this went on for 48 weeks and contained over 100 patients, there was a slowing of the rate of decline in forced vital capacity compared to placebo. So signs that treatment with pamrevlumab appears to have an anti-fibrotic effect. Now, of course, a phase 2b study is not enough for the drug to get approval. And so there are now 2 phase 3 studies being run, one of which is on no background treatment. So patients aren't taking either nintedanib or pirfenidone. In the second study, patients can be on background treatment with existing anti-fibrotic therapies. And this clinical trial is currently recruiting. It's worth saying that for people who are interested in finding out about trials running near them, this is a very useful website, clinicaltrials.gov. You can enter a number of things, including the disease you're interested in, the treatment you're interested in, and the city where you live, and it will give you trials that are nearby you. So it's a very useful resource, actually, for finding out about trials in your own area. The next drug that is in advanced development is so-called recombinant human pentraxin 2. So pentraxin is a protein in the blood that has anti-inflammatory properties and anti-fibrotic properties. And in individuals with pulmonary fibrosis, there appears to be a relative deficiency of pentraxin. And so in this study, replacement pentraxin is being given. That means that the study involves the intravenous administration of drug. This drug has been studied in a phase 2b trial. In this trial, patients were studied for 28 weeks, but then there was an open label study that continued treatment for over a year beyond that. And patients in the initial study, in the first 28 weeks, appeared to show a benefit from treatment in terms of FVC decline. So if you perhaps look on this graph here at the 28-week time point, you can see the patients on treatment had a slower rate of decline in red than the patients in placebo on blue. And then in the open label period, there appeared to be stabilization of lung function over time, although one has to be slightly cautious interpreting results in the open label phase. Importantly, the drug also appeared to have a beneficial effect on 6-minute walk. So that's a test where patients will walk up and down the corridor for 6 minutes. And what we measure is the distance that they can walk in that time. Again, if you look at the 28-week time point, individuals taking the pentraxin had completely stable 6-minute walk distances, whereas those who were on placebo had lost walk distance over the same time. And again, you can see over the longer term open label study, there seemed to be continuation of this stabilization in walk distance. And so that positive data has again been enough for the trial to move on to phase 3. And again, this study is now actively recruiting. And in this study, patients are allowed to be on background treatment with either pirfenidone or nintedanib. The next treatment to look at is a drug that was originally called TD139, renamed or relabeled GB0139. This is a so-called galectin-3 inhibitor. So galectin-3 is a molecule that is found on the surface of a number of cells, but particularly inflammatory cells, and a cell called the macrophage, which is the key inflammatory cell in the lung. We know that these cells that express galectin-3 play a role in the development of fibrosis. And we know again from preclinical models and from animal studies that blocking galectin-3 has an anti-fibrotic effect. What's particularly interesting about this drug is that it's been developed in an inhaled form. So unlike the other drugs which we've discussed so far, which are either intravenous or tablets, this one is taken by a simple inhaler in the same way that, for instance, asthmatics would take their treatment. It's been studied in a small phase 2A study. So in this study, 3 groups of 8 patients were either given placebo or increasing doses of the galectin-3 inhibitor by inhalation. What we were able to demonstrate in this study is that by using an inhaler, it was possible to get drug into the fibrotic regions of the lung. And importantly, that the drug had the expected effect, that it reduced galectin-3 levels. We also looked at blood levels of biomarkers. Biomarkers are proteins which give us some indication about the activity of the fibrotic processes in the body. And what we saw was that a number of these biomarkers, and we won't worry too much about the names, but this one is a platelet-derived growth factor. This one and this one are sort of inflammatory biomarkers that are produced by macrophages and other inflammatory cells. And what we see with all of those markers is downregulation following 8 to 10 days of treatment with the galectin-3 inhibitor. And so that's led on to a phase 2B study that's currently recruiting. So this is the only one of the studies I'm talking about, which is using an inhaled treatment. And this is now being tested worldwide. Again, they've had the bad luck of running during the COVID crisis, which has created some challenges in delivering the clinical trial. But as we get through the pandemic, the trial is now recruiting again. And again, it's available at multiple sites in the US. And then the last treatment I'm going to focus on is actually an interesting one. So it's a drug called treprostinil, which was originally developed for the treatment of pulmonary hypertension. Pulmonary hypertension describes high blood pressure in the blood vessels that flow through the lungs. And pulmonary hypertension can either occur on its own in a disease called primary pulmonary hypertension, but it frequently occurs in individuals with chronic lung disease. So it's a complication that we see in patients with pulmonary fibrosis. In the version of the disease that stands alone, the primary pulmonary hypertension, we've seen quite a lot of treatment development in the last 15 to 20 years with multiple drugs appearing to have an important effect in reducing pulmonary hypertension and reducing the blood pressure within the lungs. A number of those drugs have been trialed in patients with pulmonary hypertension related to pulmonary fibrosis. And unfortunately, almost all of those trials have been negative. However, and this is an example of one of those trials with sildenafil, otherwise known as Viagra. This drug has proven effective in primary pulmonary hypertension, but in 4 trials now in IPF, the results have been very mixed. And as a consequence, it is no longer a commonly used treatment for this indication. However, we do see that with effective treatment of pulmonary hypertension, we can improve symptoms for patients, we can improve breathlessness and oxygen levels. And so that led on to a clinical trial with treprostinil. So treprostinil is something called a prostacyclin analog. It has an effect on dilating blood vessels in the lung. It was one of the earliest treatments for patients with primary pulmonary hypertension. And so this trial was developed to see if treprostinil had an effect on pulmonary hypertension in patients with any form of fibrotic interstitial lung disease. And so the study was run for 16 weeks in patients with advanced disease and evidence of pulmonary hypertension. And the primary endpoint for this was 6-minute walk distance. And we can see here in the orange-red line that patients given treprostinil had a very clear improvement in their walk distance over the 16 weeks of the study, compared to a slight deterioration in walk distance in the patients given placebo. And so on the strength of that, the drug has actually now been approved as a treatment for pulmonary hypertension related to fibrotic lung disease. But an interesting additional observation in the study was that patients who were given treprostinil appeared to have stabilization of their lung function over the 16 weeks of the trial. And this was a slightly unexpected apparent benefit of the drug. And obviously the trial was treating pulmonary hypertension. It was working to try and improve symptoms related to pulmonary hypertension. There wasn't an expectation that this would have an antifibrotic effect. However, the slowing in the rate of decline of forced vital capacity would suggest that maybe in addition to its pulmonary hypertension effects, the drug also has a direct antifibrotic effect in patients with pulmonary fibrosis. And because of that fairly striking treatment difference that was seen in the trial, there's now an additional clinical trial being run to see whether the drug is genuinely an antifibrotic treatment in addition to being a treatment for pulmonary hypertension. And so in this study, patients with idiopathic pulmonary fibrosis at any level of severity and without pulmonary hypertension are being given treprostinil, with the goal of seeing whether it's antifibrotic in addition to its beneficial effects on pulmonary hypertension. So I've already shown you this slide before, but really it's to emphasize the point that in addition to the drugs that are in advanced phases of clinical trial development, we've also got multiple other drugs in much earlier phases of development. And so there really is hope that at least some of these are going to emerge into phase 2B and phase 3 trials in the near future. And then just one other area to think about very quickly is the treatment of not just fibrosis, but actually symptoms related to it. And so in the last couple of years, we've seen the publication of clinical trials related to oxygen therapy, and multiple trials are now going on around the world to try and better understand how we should use oxygen to the best effect in patients with pulmonary fibrosis. We've also seen clinical trials of physical therapy and pulmonary rehabilitation. And importantly, we're also seeing trials focused on treating the symptom of cough. And cough is often one of the most debilitating symptoms that patients with pulmonary fibrosis can experience. And so having run you through clinical trials very quickly in the last 40 minutes, I'll just wrap up. Clinical trials are obviously very important in developing new and safer therapies for patients with pulmonary fibrosis. I've given you an overview of the clinical trial development program so that you might better understand the language that's used. We've seen that there are multiple drugs in advanced phases of development for patients with pulmonary fibrosis. And I've also just told you that we're looking beyond treating fibrosis to treating symptoms and problems associated with it, such as low oxygen levels. So I would really just summarize by saying that we've seen huge developments in the treatment of pulmonary fibrosis in the last 10 years. Hopefully, by all of us working together in clinical trials, we can see further advances in the next decade so that we see more treatments available in clinical practice for pulmonary fibrosis. So I think there really is a lot of hope for people living with disease at the moment, because there is a huge amount that we can do to make a difference in the near future. Many thanks for listening.
Hi again. I am back with Dr. Maher. I'd like to thank you so much for that great overview of the clinical trials process and what's in the pipeline. We've got some great questions coming in from our audience, so I'm just going to hit it and we'll discuss as long as we can. I'm going to start out with a question about the conduct of clinical trials. You mentioned a couple of times the barriers that had arisen during the COVID pandemic. And I'd like to ask more generally one question and then more specific to COVID. What are the biggest barriers you see preventing patients from being enrolled in clinical trials, maybe in and outside the pandemic? And maybe as a follow-on to that, Are there things that we need to do, put in place, change about how we run and approach clinical trials, considering that the COVID pandemic is probably going to be around for a while? We're just learning to live with the problem.
Thanks, Amy. So yeah, I think with regards getting people into clinical trials, I think as a sort of specialty, we need to do better at making or enabling our patients to be aware both of trials, what it means to be in trials, and what trials are currently actively running for pulmonary fibrosis. If we look at oncology, the oncologists are very good at ensuring that clinical trials are sort of part of the potential treatment options offered to their patients. And so almost every oncology patient, once they've worked their way through standard treatment, if that has failed, they're offered a clinical trial. Whereas in pulmonary fibrosis, I think we've been a lot less good at doing that. And partly that's been due to resources. I think it's taken time to build up trial centers in pulmonary fibrosis. We tend to have fewer trial nurses working with us than oncologists do. I think some of it is, encouraging our colleagues to understand the importance of clinical trials for our patients, not just as a treatment option for the individual, but actually for the field as a whole, because without trials, we won't get new treatments. And I think we need to get better at advertising what trials are around, and that the PFF has done a good job. So, I suspect everyone who's listening in today probably knows or already knew a lot of what I said in my talk, but I think many patients are much less informed and they don't know where to go to get access to information about trials. As for the issue of continuing to run trials during COVID, I think we've learned a lot in the last 18 months. I think we've simplified protocols. So as with many things, COVID has allowed us to sort of accelerate developments in delivering closer to home care. And I think historically, we were very fastidious with clinical trials about making sure that patients came up to the hospital, that blood tests were only sent to a central laboratory. And there was a lot of bureaucracy that could have been removed and simplified. And because of COVID, that's been done. And so we now have a setup to be able to do some visits with patients at home, to send nurses out to them, to reduce the burden on the individual in the trial. And hopefully that will remain with us after COVID, because I think it has helped us to engage with patients who otherwise wouldn't have wanted to be in trials because there were just too many visits to the hospital.
That's great. Thanks. I have another question about process. Our asker says, I understand that it takes the FDA or any other regulatory agency a long time to approve a drug. And have we seen any improvements in this process since the start of the pandemic? I'm assuming this means outside of the movement that we've seen relative to COVID specifically, but in other disease states.
Yeah, so I think, I think it's a challenge that the FDA have always been aware of, and that they do get a lot of criticism, but, they have to balance on the one hand the desire to ensure that drugs are only approved when we know that they're safe and effective, with obviously the need to get drugs to patients, particularly for diseases where there's a high unmet need. And I think pulmonary fibrosis would still fall into that bracket. And if you look at sort of the history of the FDA's approach to approving drugs, the HIV pandemic actually led to a lot of changes and drove an acceleration in timelines. And I think COVID will probably end up doing the same, that there is an appreciation that it's possible to balance speed and, an appropriate appreciation of safety and efficacy without compromising, at the end of the day, what it is that comes out to patients. So I think we will see some improvements, but I think the FDA are stuck in a difficult position where they do need to undertake due diligence, and that does take a certain amount of time.
Absolutely. And I think you did a really nice job showing the amount of work that goes into all of these trials. So when we hear about something that may have come across in our email or a press release or something like that about a phase 1 study, that's still probably several years away from reaching a patient. And it's hard, to get our heads around that, but there's so much that goes into each one of those trials. And the FDA does look at all that information very carefully. And I think we need to, appreciate that, but know that they're doing the best they can in these circumstances. So I have a question about some of the agents that you talked about. What do you feel like are some of the main differences between these new possible therapies and the ones that are currently FDA approved, nintedanib and pirfenidone, in terms of not necessarily mechanism, which you talked nicely about, but is the approach to these targets different?
I think there are some differences. I think just from the mechanistic point of view, both pirfenidone and nintedanib are fairly broad acting. They're fairly sort of imprecise drugs in the sense that they don't target any one pathway specifically. They target multiple pathways at sort of lower levels of specificity. Whereas a lot of the newer treatments, the CTGF inhibitor being a good example, target one very specific pathway and do so sort of very efficiently. I think some of the other differences are around route of delivery. Our current drugs are oral. The pentraxin drug, the CTGF drug are both intravenous. The galectin drug is an inhaled drug, as is treprostinil. So I think there are also some practical differences in how the drugs are administered. And I think that may be important because at the end of the day, we're still going to keep using pirfenidone and nintedanib in the future. I think they're still going to be an important foundation to how we treat pulmonary fibrosis. And I suspect these newer drugs, if they're approved, will sit on top of those therapies. So the fact that they work in different ways and they're actually delivered in different ways, I think hopefully will ensure that these sort of future regimens of combination treatment are sort of tolerable and effective for patients with pulmonary fibrosis.
Our next, I have a couple of questions about specific treatments that you didn't cover necessarily in terms of their clinical trials, but our audience is interested in. The first is one of our listeners had a question about an early study out of Australia that looked at an inhaled version of pirfenidone, and was wondering if you knew anything about any updates on the status of that, and that it does bring up the different types of delivery and whether they might be more efficacious with one agent or another.
Yeah, so the study that's being asked about is an attempt to sort of reformulate pirfenidone so that it can be given via inhalation, with the goal, obviously, that reduces the number of side effects that the drug causes, and also the hope that it might be more effective. The company concerned have now completed a phase 2A study, which was an open label study giving patients 2 doses of drug. There's an early version of the manuscript is available on a preprint server with the Lancet, and one would anticipate that it should be published in the near future. And given that, one would hope to see the drug entering into either a phase 2B or phase 3 study in the near future.
Excellent. We have another question about an antibody to interleukin 11. I know this has made the rounds, maybe some early data on that in pulmonary fibrosis. Do you care to comment?
Yeah, so I think my name was on the press release, and I was involved in one of the preclinical studies. The study was run by a team out of Singapore with some work from London and LA involved. It suggested that blocking IL-11, which is a cytokine involved in fibrosis, was effective both in treating animals with cardiac fibrosis and animals with pulmonary fibrosis. There's been a lot of excitement about the pathway as a potential route to treating pulmonary fibrosis. As it stands, the company in Singapore has been purchased by a large pharmaceutical company who are in the process, I believe, of developing the drug further, but there aren't any ongoing clinical trials at the moment. So I would hope we will see a phase 2A study in the near future.
I know we have a lot of interest and questions about some of these early phase studies that get published or publicized. And I would want to encourage everyone listening to visit the Pulmonary Fibrosis Foundation's website, because there is a nice trial finder there, as well as a pipeline tool for drugs that are being studied in pulmonary fibrosis and related conditions. And you can you can look through that and see what is open and conducting phase 1 or later clinical trials in the United States. And I think it's a nice resource when people have questions about some of these earlier phase studies and where they might be in the process. We don't have a whole lot of time left, but there is a nice theme for questions here, asking about how we get better participation, how we get physicians to encourage patients to consider clinical trials. And maybe that gets to the idea that you were talking about of clinical trials as a treatment option for patients.
Yeah, so I think the answer to that is multifaceted. So I think this sort of talk at the PFF Summit, and I think the work that PFF is doing behind the scenes to get centers to offer trials and to drive trial participation is important. I think raising awareness of trials that are available amongst physicians and patients is important. I think we all need to lobby to increase, trial support for pulmonary diseases. I think pulmonary medicine has always been the sort of poor relation of many of the other disease areas, particularly oncology. And I think, again, if the COVID pandemic has taught us anything, it has taught us that with the right resources, I think pulmonary medicine can deliver huge amounts. So I would hope that maybe a silver lining of the sort of COVID pandemic is that there will be an increasing appreciation of the need for better treatments for patients with chronic respiratory disease. And we might therefore see some more resources in place for clinical trials and clinical trial deliveries in individual centers.
That's wonderful. Thank you so much, Dr. Maher, and thank you to everyone for your questions and your engagement today. I hope you've learned as much this week as I have. This is the last day of our live Summit sessions, but I want to remind you that you'll have access to the recorded sessions on the Summit platform through February 20th for those sessions you may have missed or want to hear again. And you'll be receiving a survey about this year's Summit on Monday. So please take a moment to fill it out and share your thoughts. We highly value your feedback and use it to guide the development of our next Summit. Thank you all so much for being here and for a wonderful week. And be well and be safe. And I'm sure we'll see you again soon.